Today I'll be talking about brain data science. In particular, I will introduce you to, I might need my slides, thanks. I will provide you with an overview of methods for neural time series data analysis. Acknowledging the type of talks that are usually done at this data fest, I feel the necessity of making several disclaiming points. So first of all, the data that I will be talking about is not an open source data. I will provide you with several sources where you could actually look for this data and then try to do with it, but just trust me, it's not an open source and it's very hard to obtain this kind of data. It's easier to go and do your experiment and obtain the data rather than find it online. Most likely you won't come back to your work and immediately apply this knowledge. So yeah, that's one more reason why this presentation might not be relevant. And also it might be so that you might never actually have a chance to work with brain data, but still I believe it's very useful. It might be very useful for you to listen to this presentation because, yeah, this is your chance to see what is brain data science, what is brain data, and how we can work with it. I also believe it's very useful to learn new kinds of data and how you could work with it. And I'm hoping that I will fascinate you with this kind of data because neuroscience needs engineers. And yeah, you will see in a moment why. Last but not least, why this presentation might be relevant is that BMI or brain-machine interfaces are becoming very popular over the last years. And in my humble opinion, I believe that the future of user interfaces are behind neural interfaces. Why I think so? Anybody knows who this monkey is? Maybe you could raise your hand. One person, that's really cool. This is the screenshot from the video that was released earlier this year by Elon Musk's company Neuralink. What this monkey is able to do is that he or she can play the game of Pong just with the use of her brain. So she has an electrode, or he has an electrode, and she controls the gamepad just with her brain. And I think this is a huge advancement, but not in terms of the kind of task that the monkey is able to do, because neuroscientists were able to do this kind of things for decades, but with the engineering aspect of it. So Elon Musk and his company, they were able to pack everything very densely within a very tiny electrode. So this is where the really cool thing, where the advancement of this technology is. You probably know who this guy is. That's the founder of Valve, the huge game company. And he also earlier this year released his plans for the neural interfaces. And in this presentation, that's also a screenshot from the presentation from an interview, and he was saying there that he's not only looking forward to create neural interfaces that could read from your brain so you could control the computer, but could also give you some new kind of senses. As a neuroscientist, I'm really interested how he's going to do that, but probably we will see that in the next couple of years. So a little bit about me. I'm a psychologist and software engineer by training. Once upon a time, I also did a FORSET fellowship. FORSET is an organization that organizes whole data fest. Currently, I am working on my master's research in cognitive science in Germany. And yeah, I'm working as a research assistant with neural and behavioral data. On the left, you could see just one example of the kind of visualization that I was working with recently, and that's me participating in an experiment with the EEG cap. Yeah, so introduction. There are many ways how you could measure the brain activity. All of them have their own advantages and disadvantages. Let me go through this very busy graph together with you. So on the vertical axis, you could see the spatial dimension, and on the horizontal axis, you could see the temporal dimension, what that actually means. So the spatial dimension means how huge of the scale you could measure the brain. For example, on the scale of the entire lobe, so that's one part of lobe, that's another part of lobe, and then here is another part of lobe. When you go narrower, deeper, so to say, you could measure neurons, dendrites, and synapses. And smaller is the scale, the better kind of information you could get. So the more precise on the single cell level. If you go on the temporal resolution and see what you could do there is that, for example, fMRI, it measures, it's very slow measurement technique. It updates every several seconds, but brain processes are transient events, and you really need a very high update rate in order to really measure the processes on their real temporal scale, so to say. And that is why I believe EEG and MEG are really cool kind of techniques, because they measure the brain processes at the real scale, in real time, so to say. For example, if there's a spike, it measures in the temporal dimension exactly as these processes are happening. And my presentation will entirely be about EEG, and I will slightly cover what is MEG and how it is different from EEG. Yeah, what is EEG basically? It's electroencephalography. It was discovered a bit less than 100 years ago by Hans Berger. And the first kind of waves, the first kind of signal that he was able to record is the alpha waves from 7 to 13 hertz. You will get a bit more in detail what are the frequency ranges and all that kind of stuff. So basically, how does the EEG experiment happen is that basically you attach this EEG cap to the head of your participant, of your subject, and simultaneously you record the behavioral responses, for example, if there is a button press or something like that, and the EEG signal. And then you could kind of relate the behavioral responses with the EEG. But there is one exclusion from this, I would say, that's exception. There are resting state studies when the person does absolutely nothing, and then still, like neuroscientists measure what kind of brain activity might happen in a resting state. So person does nothing, and then still we could see some activation. Yeah, and after we record the data, we could see some, the signal might look somehow like this. So on the y-axis, you see the electrodes, and on the x-axis, there is this time. So as it goes across the time, you see how does this fluctuation happen. So what is basically EEG and what does it measure? So EEG measures electrical field created by simultaneous activations of population of neurons. So basically you attach the electrode on top of the scalp, and through volume conduction there are some, so down there you see where the arrows, that's the brain actually, that's some of the lobes of the brain, and through volume conduction through scalp and through skull itself, you could measure those processes that are happening inside of your brain. MEG is slightly different. It measures also the activations of neurons, but it measures the magnetic properties of the activation, so not electrical properties. So when neurons and when some particular parts of the brain are activated, there is a whole lot of different processes that are happening, such as blood inflow, which is measured by fMRI, and some electrical fields that are generated, that is measured with electrophysiology. It's Neuroscience 101, just so you know what exactly we are measuring. So there was a huge debate regarding electrophysiology, but most of the neuroscientists agree that what we measure here is the movement of electrical charges and electrical field. But I will go into that in a moment. So on the left, over there, way back to there, you see the representation of a neuronal cell. Neuronal cell has axons and dendrites, so it takes an input and it produces an output, same as your artificial neural networks. And when this signal propagates from dendrites to axons, it generates this electrical field, as you see in the middle. Yeah, and when there is a synchronization, for example, when there are thousands or tens of thousands neurons are active at the same time, we could measure that activity because the electrical field that is generated by this biological neurons is very high. It also generates a very high magnetic field that can be measured with MEG. And yeah, so simultaneous activation of population of neurons oriented in the same direction causes LFP. And you could see that arrow over there, for example, when all of the neurons are directed in the same way, we could measure that with the EEG. Yeah, so let's assume you conducted your experiment, you recorded the data might look like this. But actually I'm lying here a little bit. It's very unlikely that you will get this kind of clean data. I feel like this was like thoroughly pre-processed, and in a moment we will see how it is done. The kind of data that you might get after your experiment might look like this. This is a bit exaggerated example. It's very rare that you get this kind of example as well, but still you might, I'm showing this just to motivate why my presentation is relevant and why you need all the preprocessing steps. And how to go from this to this. This is the gist of my presentation, and you will see that there are several major sources of noise in EEG. It's an eye movement, muscle movement, so muscle activity, heart, line noise, and channel noise. Line noise and channel noise are hardware-related noise, and all of the first three are related to biology-related noise. So what is an eye movement noise? When you move the eyes, for example, left to right, there is a huge saccades that you could see on the graph back here. There is an immediate kind of oscillation that you will see because those muscles are very strong, and you could see how your eyes are moving in the EEG signal. Also, when you blink, there is, again, this oscillation that is very strong and that could kind of spoil your data, so to say. That's why it is usually recommended to have a fixation point so participants are not moving their eyes too often. And also sometimes people request their participants to not blink too often. But then that's another issue, because when you request your subjects to not blink too often, they might kind of keep that information in their working memory, and that might cause some other kind of noise, but that's out of scope of this presentation. And muscles as well. For example, when you kind of twist your eyebrows, that could create, I don't know, the noise that would look exactly like this. That's a very strong kind of noise that you would see on the EEG signal. Yeah, and the entire talk in one sentence is just the brain data is noisy and you need to go through a lot of cleaning before it becomes usable. So yeah, now let's dive deeper. What do we need to do in order to get a clean EEG data? So yeah, these are the 9 standard steps in EEG data preprocessing that you need to go through. But for the sake of time, I'm going to omit the first and the last one, and also the fourth one, because I will need to go through a lot of, I will need to basically provide you a lot of information so you kind of understand what is re-referencing. Yeah, so let's start. What is downsampling? All of these measurements have their sampling rate, so that is measured in hertz or SPS, samples per second. And the reason why you want to do downsampling is that you want to reduce your data size, because maybe you recorded your data at a very high sampling rate, but then you don't really need it at such a high sampling rate. And another reason why you might also want to do downsampling is that it will significantly improve your computation speed, because there are some algorithms that takes, I don't know, several hours at least. For example, ICA, that we will go to later on. A normal computer, it would take like, I don't know, at least 50 minutes to 1 hour was the normal data set. So typical sampling rates are 500, 1,000, and 2,000 samples per second. Also, you might kind of meet the kind of sampling rates that are not really recommended. So it's 512, 2024, and all other powers of 2. The reason why it is not recommended to do downsampling or do the sampling rate at those sampling rates is because it's very difficult to relate the time and the sample rates per second. Because when you have 1,000, for example, one time point is exactly one millisecond. But if you do that with 1,024 samples per second, it's very difficult to relate. I don't know how, honestly, it's very hard to convert from 1,024 samples per second to milliseconds. Important notes that you should consider when you are doing downsampling is that you need to really know what is the maximum frequency that you are interested in, because of the Nyquist theorem. That suggests that you could not look at the frequencies that are 2 times, so the minimum requirement is 2x of maximum frequency of interest. You could see that on a sine wave here, for example, if you're sampling the sine wave, and if you measure, if you're sampling at less than 2 times the frequency, you might get something like a flat line, though it was a sine wave. So you could undersample your data. And again, this is oversampling your data because it measured at the 20x of the highest frequency, of a frequency. And it's not really recommended because there wouldn't be a high difference between 10 times or 20 times the frequency. So that's sort of oversampling, and you might want to get rid of that. Filtering, that's the next step. After you do the downsampling, then you might want to do filtering of the signal. So you need to remove high or low frequency artifacts, because those eye movements or muscle movements are usually on a slow oscillatory wave or on a very high oscillatory wave frequency range, so to say. And you want to get rid of it as well. So there is a high-pass filter, low-pass filter, and band-pass filter. So what does the high-pass filter do? It retains only high frequencies. By analogy, the low-pass filter retains only low frequencies. For example, you could go from 0 to 200 hertz and retain only those frequencies. Also, if you know exactly at which frequency range is some phenomena happening that is of your interest, you could do the band-pass filtering. For example, you could narrow down your data to gamma band, and then you might be interested only from 30 to 80 hertz, because gamma band on average is somewhere around there. But if you look at the literature, those bands are not precise. Some people define it as 30 to 80, but you might encounter some other frequency bands. Again, an important note here, so the narrower the band, the less is the temporal precision and more temporal smoothing. What does it mean exactly? If you take a very narrow band, you might not be able to tell when exactly in time some particular event happened. That's because of the algorithms that are applied for filtering. And basically, for example, if you are talking about millisecond range, if you have a very narrow band, you won't be able to take it such a precision in time. This is how the signal might look like. You see on the in the signal above, so there is a very slow oscillation that is happening. So you could see a very slow sine wave and also very transient sine wave. So it goes very sharp. And after filtering the signal, it becomes more or less clean. I mean, much cleaner than it is above. And like I said, you could choose a very specific frequency band, and you could extract some particular delta wave or theta wave or alpha wave. It depends on your experiment and on what kind of wave you're interested in. But I will get into that in a moment, how you could know what kind of wave you're interested in and all that kind of stuff. So that's just general information about EEG frequency bands. So we talk about the first 4 steps. Now it's probably getting more and more interesting. So how you inspect electrodes and reject noisy channels. So after you've done some filtering, you might get some data that looks like this. And you see that the channel P7, but it's not the only kind of channel that is very noisy. You see that there is a very high oscillation that goes very beyond its own line. You see that it's a very noisy channel. Maybe it was detached from the scalp. It also happens that the cap moved slightly and the electrode is not attached directly to the scalp. So you might just reject it, get rid of this electrode. So it's not going anywhere further to the analysis, because that's just a noise. You won't be able to do anything with this data. So you just look at it, identify it, and just get rid of it. Of course, there are some methods how you could quantify it, but again, that's out of the scope of this presentation. In order to proceed, and so you could understand the next point of preprocessing, I need to go a bit deeper into EEG experiment. So on the example of N-back working memory paradigm. So what is happening here is that the sequence of letters are presented to a subject, and a subject has to press the button whenever the letters are repeating after another letter. So for example, you see with the letter B goes, then Y goes, and then B goes. And as soon as the second B has appeared on the screen, the subject has to press the button because there was a repetition back. And what the subject actually has to do constantly, the subject constantly has to update his or her working memory contents. So for example, R appeared and subject goes like R, and then B appeared, subject goes RB, RB, RB. Then Y appeared, subject goes BY, BY, BY. And then as soon as the next B appeared, you kind of could recall that there was 2 back, that's repetition, and then you press the button. What we do simultaneously, we could record the EEG data. So the kind of neural data I was talking previously, and behavioral data. So when exactly did the participant press the button? And also what we record is when exactly in time did the image appear on the screen. So this is a very important information because we will need that for the experiment. Yeah. Yeah, and what you could see on the y-axis here is that it's not a continuous time, it's kind of done in a chunks. What do those chunks mean? So it's the data that you see here is centered around 0. And 0 is an event that was recorded from the behavioral kind of data. For example, if you, the zero event could be either the presentation of an image or when the button press has happened. And you could identify and kind of separate those chunks. Yeah. And you immediately see that a couple of milliseconds after the stimulus was presented, so after a couple of milliseconds after the 0, there is a strong kind of, yeah, something that looks differently from the rest of the data, so to say. And yeah, that could already give you some hints to consecutive analysis. But it might not always look as good as here. This is actually an exceptional case that I'm showing throughout multiple times during my presentation. That's very clean data, I would say. The kind of epochs, those actually chunks of time points are called epochs or trials. Some of the epochs might be very noisy because maybe a person moved, or I don't know, maybe there was a itch and then the person scratched a little bit, and that would create a lot of noise. Even if there is a hand movement, that would also create a lot of noise. And we just identify those bad epochs and we just get rid of it. So yeah, so we've done mostly with most of the preprocessing steps. Now it's time for running independent component analysis and rejecting noisy components. Independent component analysis, or ICA, is better understood through cocktail party problem. So imagine you are in a cocktail party like you were during the lunch, and then there is a lot of noise. So a lot of people are talking simultaneously, someone, I don't know, making noise with the fork, or I don't know, many noises could appear on a cocktail party, and you are trying to figure out someone's particular speech. So you are trying to hear that your friend or colleague is talking, and how you could do that, you kind of put several microphones, but not one, because ICA wouldn't run when you have only one recording device. So you need to have several recording devices, and you feed this data into ICA, and what it does, it learns the unmixing metrics, and it kind of unmixes the data into independent sources. So it finds unmixing metrics based on statistical properties of the data. And there are several examples online that you could just Google ICA and cocktail party problem, and you will hear that there is a very noisy sound was at the beginning, but after we run the ICA, there is a distinct speech that you could kind of identify and hear what the person is talking about. The requirement of ICA is that the data that is fed to ICA should be as clean as possible, because if you feed into it very bad data, you will get a very bad data at the very end. So it is very important to do all those steps that I talked about previously before running ICA. Yeah, so when we do ICA on EEG data, we could end up with kind of topographical distribution. Yeah, we could end up with having several independent components. And one important note here as well is that the number of independent components should always be less than the number of electrodes, because it's very unlikely that you will have the number of independent sources that is more than the number of channels that you have. Yeah, you could see the topographical distribution, but that's, I'm showing a window of EEG Lab. That's the MATLAB kind of plugin that is very commonly used for EEG data. And when you click on those topographical representations, you could actually get, oops, yeah, maybe I will talk about it in a moment. So yeah, I wanted to say that the independent component number 1, and I'm zooming it in here, you see that it was able to identify eye movement related components. So those are very prominently, you can see that those are eyes, and immediately you want to reject it. And you kind of go through this topographical representations, and maybe you could also look at this activity power spectrum and also ERP plot or ERP plot that you see over here. And if you are a neuroscientist or you have worked with a lot of EEG data, you might immediately know which of independent components are the artifactual. Artifacts are just the noise of the EEG, so to say. But imagine if you're having tens of hundreds of subjects, or if you're not a neuroscientist here and you don't know how to identify which independent components are artifactual, you could actually automate this process. And there are many ways how you could automate it, but I am particularly amazed by the recent development in this field. So in 2019, they created this IC label, and what it does is it is a multimodal deep neural network that gets all of this information, and it kind of gives a probability whether this independent components were artifactual or brain-related, and it gives you these probabilities. There are 7 classes that IC label can identify. It's brain, eye, muscle, heart, line noise, channel noise, and other. You could see that independent component number 4 has a very high brain probability, and indeed, if you are a neuroscientist and you look at topographical representation, it really looks like a brain activity, unlike the independent component number 1 that I showed previously, which has a 99% of being an eye component. Yeah, so imagine that you kind of have these probabilities, you know which probabilities, which independent components you want to reject. So how does this rejection actually happening? So I told you previously that if there is a blink or eye movement or any kind of eye artifact, you would see this kind of shape prominently on the EEG graph, so to say. And yeah, what you do is that you subtract this independent component signal from the real signal. And at the very end, you could see that red squiggly kind of line, that's the end signal that you are having when you subtract the independent component from the true signal. Yeah. And now we finally can tell that the data that we obtained from the experiment actually looks as clean as I showed you somewhere at the very beginning. After applying, I mean, most likely after applying, you won't get exactly like this, but it would be somewhere close to this one. All right, so you have the clean EEG data, and what you could do next is you could answer questions with this EEG data. Yeah, so we cleaned it, so what's next? Now starts the most exciting part. And there are two main approaches how we could answer questions with the EEG data. But there's a myriad, there's a whole ocean of different methods that I'm not talking here, but this is just the main ones. So ERP, it's a univariate approach, it's event-related potential, and multivariate approach, which is a multivariate pattern analysis, or MVPA. What MVPA is, you basically train the machine learning model based on those EEG time series, and you can predict whether there is some contents in the brain of a person based on the EEG data. I'll get into it in a moment. So what is ERP? That's just a very brief introduction to ERP. There are whole volumes of books that are devoted only to this technique, so it's not an extensive introduction to ERP, I would say. So what you do is that you localize the activity of interest based on topographical distribution. For example, you are interested what happens at 100 milliseconds after the stimulus was presented. You kind of take that piece of data, you kind of average across the trials, and you could get this kind of topographical distribution. And you could see somewhere a bit frontoparietal region, somewhere over here, you could see there is a very high activation. So you might be interested exactly in that electrode or in electrodes surrounded there. So you take only one electrode from all of the electrodes that you have, and you kind of average across trials. So when you superimpose all the trials that are centered around 0, as you see over there, you might see that there is kind of there's a prominent kind of squiggly line that happens in most of the trials. If you average it, you could get something like you see on the right, but actually, again, that's not totally true, because in neuroscience, it comes from the physics. I guess it's that's how they actually do this y-axis of most of the plots, so it's inverted. So the negative numbers goes to the up, and then the positive goes to the right. And then when you invert it, you see that most of the trials are represented as N1 component, and then there is also, you could see the P3 component over there. N1 is auditory stimulus perception component, and P3 is decision making. Some people done a research, and when there was kind of decision making or auditory perception task, they realized that at, I don't know, 100 milliseconds after the stimulus was presented, there is this N1 component. Now it gets to something more interesting, multivariate pattern analysis, or MVPA. And yeah, so you could train machine learning models on this data, and there are many approaches how you could actually pre-process your data in such a way so you kind of can feed it into machine learning models. So the first approach is that you could take the topographical maps that I showed previously, you take those topographical maps and you feed it into machine learning model. Another approach that you could do is that you could actually convolve across time with 1D convolutional nets across all of the electrodes, and then you could see maybe, yeah, you could maybe be able to decode some information from there. Another approach, and that's, I believe they achieved one of the highest performance, but it's very difficult to do this kind of things in real time. So for example, if you have a real-time brain-machine interface, you most likely won't be able to do 2D convolution. So what you do is that you pre-process the data into this kind of scalograms, where on the vertical axis there are frequencies, and on horizontal axis there is time. And you kind of feed this into 2D convnet, and it kind of also can pick up on this image and be able to decode what might be represented in the EEG signal, so to say. All right, so you learned something, and I believe it's very useful for you to kind of apply knowledge. Otherwise, what's the point of this whole thing? So what you could do immediately after this talk, you could go to Google or to Kaggle, and you could type EEG, and that's one of the few ways how you could get hands-on EEG data, so to say. You just type EEG, and then you see that there are several notebooks, several codes, and even 18 competitions related to EEG. Trust me, most of them are in-class competitions, so you won't be able to earn a lot of money, and those that were not in-class competitions are already over for many years. Yeah, so data is not open source, but there is Kaggle. For this presentation, I wanted to provide you with a very simple kind of competition that was there last year. I think it's done with Yandex, if I'm not wrong, with this NeuroML 2020 kind of summer school. What they were doing is that they were taking the EEG data and they were decoding whether there was movement or resting state. The metric for this competition was ROC AUC, or ROC AUC, and there is a binary classification. So what you decode is whether there is a movement or there is no movement. In the data, you could see there are 19 electrodes and 14,580 time points in train set, and 9,000 and something in the test set. If you a quick Google search, what kind of brain area might be related to the motor processes and kind of movement. Since we are decoding movement, you might be interested in that. You Google, and immediately the first response is that the frequency band of your interest might be from 60 to 80 hertz. And the electrodes that are located somewhere here on the side and here on the side, so these are the motor cortexes over here. So the electrodes that you might be interested in are located somewhere here. So it's central electrode, central and to the side. But I need to tell you the truth that this data is it's a toy data set, and you won't be able to do any kind of filtering and all the kind of preprocessing that I was talking to you about previously, because yeah, this is pre-cleaned and it's given to you in a very nice way, so you don't need to care about all this preprocessing. And like in real life, yeah, as you know that in Kaggle everything is very toy-like. So yeah, you could immediately plot one of the examples from frontal electrode. So you see F4 over here, it's a frontal electrode, and there are two conditions, for example, hand movement and the resting state. And you immediately can visually see that those conditions are differently represented on the on this plot. And what you could do immediately, and what they suggest in the baseline, is that I think either SVM or logistical regression based on the data from only one electrode. So you have 19 electrodes, you throw away the data from 18 electrodes, and you're interested only in one electrode. But I think it's not the best approach, and because you have a lot of data and you're just ending up with only one kind of data, maybe there's more information that you could get from other electrodes. So what you could do is you could train 1D convolutional network on completely raw data, but this raw data is kind of pre-processed. So here you could do that. So the data batch looks like n samples, n time points, and n electrodes. And what it does, it actually, this 1D convolutional networks, it takes one time point and it convolves through all of the electrodes, and it kind of, so it kind of learns which features might be more relevant to distinguishing between these two classes. Yeah. But again, like I said, this is a very toy example. There is another competition that was 6 years ago, and there was a prize of $10,000. And what you do there is that you need to segment the time series, this EEG time series, into several conditions. So they had a hand movement, resting state, and I think 5 more classes. And what you need to do is you kind of need to predict whether at some particular time point there was a movement and when it ended. So you kind of need to segment this temporal data. So conclusion, if you want to go kind of deeper, there are several libraries how you could do that. In Python, there are only 2 because neuroscientists, unfortunately, are not using Python, and there are only 2 libraries for doing that, MNE and NILearn. NILearn is a neuroimaging learn. It's very similar to sklearn. If you're familiar with it, you will be able to pick it up very quickly. I think it's even written in the same fashion as sklearn. But most of the neuroscience research is done in MATLAB, unfortunately, and the kind of libraries and plugins that you could use if you're using MATLAB are EEG Lab and Filtrip. So this is not the very end of the presentation, but if you want to dive deeper, most of the things that I was talking about were taken from this book, but not exclusively. If you want to dive deeper, you could find this book somewhere and read how you could analyze the neural time series data. Also, I recently started the GitHub Pages page, so to say, and I will start pushing some of the work that I am doing at my research center soon. So there is some of the things that are already there, but then you could find also my contact information there. So I would really recommend you to try this competition yourself, and if you have any questions, it's time for you to ask them. Thank you. Yep.
Then, if you would analyze my brain now, I think you will find interesting patterns because my brain gave error because of this presentation. I can hardly say that I understood many things, but the question is what this data is used for.
Used for?
Yes.
So this data, mainly in the neuroscience research, it's used for studying how does the certain processes or certain phenomena represented on a neural level, on a population of neurons. For example, we don't know what happens when you do an error. For example, you want to say something, but then you kind of are not sure whether the kind of word that you're saying is exactly, so there's kind of a feeling of not being sure that the word you're saying is exactly right. So you kind of start doubting. Also, when you are giving an answer to a question, you again might be doubting what kind of answer you are giving. Maybe I am also having this doubt at the moment. So if you want to study how this happens on the neural level, you kind of wear this EEG cap to your participant. You kind of create this environment. So there is so the person kind of commits or does an error several times during the experiment, and you learn what happens. And people already study this. There is something like error-related negativity, and you know that several milliseconds after an error has been made, there is some negative deflection in the central electrodes somewhere over here in the EEG signal, and you know how it actually happens in the brain. So answering your question very shortly, this is done to study the brain, but in the BMI, it is done to kind of decode and For example, there are people who are paralyzed, and it kind of helps those people to control the, I don't know, maybe prosthetic arm with their brain. So there are many ways where you could use it, but in neuroscience, it's used for, yeah, for science purposes, just to know how does the brain work and all that kind of stuff. Any more questions?
I think there are no more questions, so thank you so much.
Thank you.